Welcome to this lecture series on eukaryotic gene expression, basics and benefits. Today we are on the lecture number 39. We are primarily going to discuss about a very interesting and very novel topic, genomics and proteomics. Now the purpose of giving this lecture is to make you understand how we can study global changes in gene expression, how we can understand global changes that take place in gene expression, what are the new technologies that are available to us, and what are the new challenges ahead of us. Now, the year 2000-2001 is a kind of a landmark in the molecular biology as well as gene expression and eukaryotic genome biology, because the complete genome sequence of the humans was published by two independent groups, one that appeared in Science, another appeared in Nature. So this sequencing of the human genome told us about the number of genes that are organized in the human genome and paved way for sequencing a number of other genomes as well. So we are basically in a post-sequencing era where sequencing genomes of several organisms has provided a wealth of information leading to the creation of several new disciplines, be it microarray based disciplines, bioinformatics, genetics, to just name a few. Now what has become evident once the genome sequence became available not only for humans but also for many mammalian species such as mouse, rat and so on and so forth, about 40 to 60% of all the genes that were identified were classified as unannotated genes, because we do not know what the function of genes is. We do not know what kind of proteins they code for, and we do not know anything about the function of these proteins. So one of the important things that came out of this human genome sequencing or sequencing of mammalian genomes is that, or even microbial genomes, that we still do not understand the function of many of these genes. So with more and more sequencing technologies becoming available and more refinement of these technologies, more and more genome sequences are being made available, and it is actually estimated that in the GenBank, which is actually a depository for all these such genome sequences, the sequence data almost doubles every 18 months, and about 190 billion bases of sequence data is being added to this GenBank almost every year. And there are approximately about 200 eukaryotic genomes and about 600 prokaryotic genomes either being sequenced or close to completion. So what you can see, what is happening is that a huge volume of data is being generated, and to actually make sense out of all this data, it became impossible for a human to handle all this huge volume of data. So a new era of computational biology known as bioinformatics came into prominence. So you required softwares to be developed, you required computations aids, you required computers to handle this huge volume of data and organize so that one can actually go and retrieve the information that you are interested in. So let us now just take a look at what the human genome sequence data told us. The human genome data sequence actually told us that only 1.1 to 1.4% of the human genome DNA actually codes for the proteins. That means only a very small fraction of the human genome actually is involved in coding for proteins. There contains the protein coding genes, as I can shown here. So only 1.1 to 1.4% of this entire genome is actually translated into proteins. And very surprisingly, almost 25% of this human genome actually codes for RNA, which never gets translated into proteins. So the end product of this genome is actually RNA. And we have studied in our previous classes that how RNA can have very important regulatory functions like the microRNAs, siRNAs and so on and so forth. In addition to the small RNAs, there are also a number of other RNAs which are broadly known as the non-coding RNAs, because they do not code for any proteins, and the RNA itself is the final functional entity. So a lot of information is coming out about the structural or functional or regulatory roles of these various RNAs, which are the ultimate end products of a huge part of the human genomes. And it turned out about 3% of the human genome consists of what are called as microsatellites, which became very important for identification of disease markers and for what is called as a functional genomics. And also some known to contain large duplications and in many other regions of DNA, which is broadly classified as intergenic DNA, And very interestingly, about 45% of the human genome sequence consists of what are called as moving elements or transposons, which can jump from one place to another. So as you can see, what the human genome actually told is that people who have been focusing their attention all the time on protein coding genes and how RNA polymerase to transcribe genes, how messenger RNA is synthesized, they all became annoyed with this kind of information, saying that what they are actually studying is only a very small fraction of the human genome, and the rest of them probably we do not know anything about. So if you now look at these about 30,000 protein coding genes, that is what is the estimate in the case of the human genome. All these 30,000 protein coding genes are not expressed in any given cell type, and it is estimated that about 18,000 transcripts may be present, and therefore these 18,000 transcripts may be translated into about 18,000 proteins. This is the approximate estimate in about an average number of transcripts that may be present in a given cell type. Now among these 18,000 proteins, about 2,500 may be common to all cell types. These are what are called the housekeeping proteins, which are required they are involved in energy metabolism or structural components and so on and so forth, and about 2,000 may be secreted out. So if you now deduct this from 18,000, approximately 13,000 genes may be cell specific, and assuming that there are about 300 different cell types in various mammalian systems, there are about 45 to 50 proteins that may actually be highly cell type specific. So these are all the average estimates that one gets out of the information that is available in the human genome databases. So you have a human genome which approximately codes for about 30,000 protein coding genes, and these 30,000 genes, by a number of other processes such as differential transcription initiation, differential splicing, or differential polyadenylation and so on and so forth, can actually generate multiple transcripts, because we have already studied that how more than one messenger RNA species can emanate from a single gene. So it is estimated that about the human transcriptome, that is the number of RNA molecules that can be synthesized from the human genome, is anywhere varies from 40,000 to 100,000. So this many messenger RNAs can be synthesized, or the human genome has the potential to synthesize this many number of messenger RNAs. Now again, when this messenger RNA gets translated into proteins, again proteins can undergo splicing, or they can undergo various post-translational modifications, and each post-translational modification can give a different function to the protein. So the proteome, that is the protein component of the human genome, actually consists of anywhere between 100,000 to 400,000 proteins, which can have different functions. And these proteins, again, they are not in the cell. These proteins interact with each other and form key components of either metabolic reactions in the form of enzyme complexes or major signal transducers in major signal transduction pathways, and these protein-protein interactions may almost be around 1 million. So you can see the ultimate physiology of an organism, of a mammal, ultimately depends on these 1 million protein-protein interactions that ultimately govern the various functions of the cell or the organism. So you have genome, and you have transcriptome, you have proteome, and these proteins interact to generate what is called as a interactome. So as you can see, from the human genome sequence, it became very clear, as you come to the functional side of the mammal, the number increases logarithmically. So we have now basically entered what is known as the omics era. I just now explained the analysis of the information, sequence information in the genome give rise to what is called as a genomics. And when you start looking at how these genes are transcribed into messenger RNA, and then look at the diversity of these messenger RNAs, you get what is called a transcriptome or a transcriptomics. And then when the messenger RNA gets translated to proteins, you want to look at the various variations in proteins and how these proteins get modified and how they contribute to the function. You get what is called a proteome or a proteomics, and when these proteins interact with each other and form interacting networks and then contribute to specific metabolic pathways or signal transduction pathways, you get what is called say metabolome or metabolomics. So as we go from genome to metabolome, the complexity increases several fold. Within genomics, again several sub-disciplines came into being. I have just listed a few here. You have what is called as a functional genomics, which describes the way in which genes and their products, that is the proteins, interact together in complex networks in living cells. And if you disrupt these interactions, it can lead to a number of diseases. So functional interactions between genes and gene products leading to normal physiology, disruption, or how these disruption of these interactions can lead to disease phenotypes, what is known as a functional genomics. And you have structural genomics, which primarily focuses on architectural features of genes and chromosomes, how these genes are organized, euchromatin, heterochromatin, the clusters, intron exon organization, and so on and so forth, the repeat sequences and all these things. Then you have what is called comparative genomics, because as more and more genome sequences became available from a number of organisms, people started comparing. For example, if you compare a human genome with a chimpanzee genome, it is more than 99.5 or more than 99% identical. So what distinguishes between a chimpanzee genome and human genome is only 1%. So you can really see these variations that we see between organisms or between species may not entirely be accounted for the sequence diversity. So we need to come down further towards proteins and their functions and the RNA and their functions to really understand how this genome ultimately contributes to diversity among various organisms. So I have comparative genomics, which basically looks at evolutionary relationships between genes and proteins of different species. And epigenomics, which I have studied in great detail in the last few lectures, which basically talks about genetic effects not actually caused by changing in the DNA sequences, epigenome, that is above genome sequence. And usually these kinds of epigenomic changes involve either DNA methylation or histone modifications. We have studied extensively how epigenetics plays a very important role in number of diseases as well as development. Then you have what is called pharmacogenomics, that is how you can make use of this genome information and the proteome information to design new drugs or new vaccines, how you can develop new drug targets, how you can develop new drugs, and so on and so forth. So you can see the sequencing of genomes has really opened up whole new areas of research in the area of biology, and a number of new disciplines came into being to really understand what is going on inside the cell and how all these transcripts, proteins and the genes contribute to the diversity among organisms. So the genome sequence basically tells you about what can happen. Now once the genome, the RNA gets made, or once the transcripts are generated from genome, the transcriptome basically tells you what appears to be happening. Now once these RNA gets translated into proteins and you start looking at the proteome, you understand, because proteins are the workhorses of the cell, it actually tells you what makes it happen. And then when ultimately these proteins work together, interact with each other, generating networks, and you start looking at metabolic interactions and metabolic networks, you actually realize what actually happens. So in order to understand, to connect this genome information to the metabolome, you require key players, and one is called the bioinformatics. Huge amount of computing power is required to put together all this data and make connections between what is happening at the gene level and what is happening at the metabolism level. And also a very new area that actually does this, uses the computing power or uses computational biology and the sequence information and the various experimental data available, is actually what is known as a systems biology. So very complex areas of research and has actually come into being with the analysis of the genomes, proteomes, and so on and so forth. So let us just spend some time to now understand what actually is going on in the area of genomics. What are the exciting developments that are taking place in the area of genomics? Now what is genomics? A simple straightforward definition of genomics would be rapid identification of all the genes expressed in a cell or a tissue. This is the simplest definition of genomics. How will you quickly identify that what are the total genes, total number of genes that are expressed in a given cell? Now so far in all our studies earlier, we have been studying only gene expression changes that are happening at the gene level. At the most we are looking at a cellular level, at different stages of development, and how each transcription factor or how each gene contributes to certain important changes. But today with technology has developed and technology has made it possible to understand all kinds of genes that are expressed, that is the total changes that takes place in a genome at the level of transcription, and you can look at the entire gene expression change that is taking place either at the cellular level or at the tissue level. The key player for that enables us to understand this kind of a global changes in gene expression that is taking place in a cell or in a tissue is what is called as a DNA microarray. So let us spend some time to understand how actually one can do genomics by using these microarrays. What is a microarray? A microarray is nothing but a small 1 square centimeter chip that is divided into several thousands of small squares. Now each square contains many copies of a single gene, and this microarray was initially developed by Patrick Brown at the Stanford University School of Medicine to determine which genes are actually involved in each cell sporulation. Let us spend a little bit more time to understand this microarray, because that is the key player for understanding for doing genomics. So what happens with the DNA microarrays? DNA microarrays allow you to look at expression of all potential messenger RNAs in a cell at the same time. Now we have earlier studied techniques like RT-PCR, Northern blot, and so on and so forth, where at the most you can look at a handful of genes, maybe 10 genes, 20 genes. You can look at the number of genes that are expressed in a particular tissue, but the number is limited. You cannot go beyond 10 or 20 genes. But if you want to look at all the genes which are getting expressed in a given cell, you can only do it with technologies like microarray. So what actually happens? A microarray, as I said, is nothing but a chip which is actually present on a glass slide. They are composed of short DNA oligomers attached to inert substrate or a glass slide. Each dot that you see here represents one particular gene probe. So let us say if there are about 30,000 spots here, that means 30,000 individual genes that have been laid on this microarray. Now typically each one of this grid consists of about 105 to 106 spots, each with a different DNA molecule. So these DNAs are actually spotted onto this glass slide by using a technique called as lithography. Now many a times in earlier times, people usually actually synthesize this DNA either in the form of oligonucleotides or cDNAs, and then this used to be spotted onto these microarrays by robotics. But today it is actually possible to do the entire gene synthesis right on the chip. So you can actually synthesize different DNA molecules right on the chip and using what are called as oligonucleotide arrays. So once you have all these genes which have been spotted on these DNA microarrays, you now take this microarray. For example, you call it as a mouse microarray when you spot all the genes present in the mouse genome, or oligonucleotides corresponding to different genes of the mouse, or you call it as a human microarray if human genes are spotted, and so on and so forth. So once you have these kinds of microarrays ready, you then hybridize these chips to fluorescently labeled DNA and RNA, so which are complementary. So you can isolate, for example, if you want to look at RNA which is expressed in mouse liver under two different conditions, you extract this RNA and then label them differentially using two different dyes and hybridize to this particular chip, and then you wash off the unhybridized probe and then scan this using a laser scanner. I would not like to go into too much details of this thing because it is a very fascinating technology. I would actually suggest you to actually look at the animation of DNA microarray methodology, how actually these microarrays are developed and how the microarray experiments are carried out. There is a very nice animation. You can go to this particular website, either this or this, just cut and paste it, and then just look at a beautiful video that actually tells you how actually these microarrays are made and how actually a microarray experiment can be carried out. So I am just going to very briefly tell you how exactly a microarray experiment is done. The rest you can go and watch that video. Let us assume, for example, there are two different conditions. One is a normal condition, another is an experimental condition. This can be anything. For example, you want to now, for example, look at a normal cell, what happens to gene expression in a normal cell versus what happens to gene expression in a cancer cell. Or you can look at what is happening between a unstimulated cell and a stimulated cell. Stimulated cell, I mean you can add a hormone or you can add a growth factor, any component that you are interested in. So any two different physiological situations one can take, and then you basically isolate RNA from these two different conditions. So you isolate RNA from the normal cells, or you isolate RNA from your whatever condition that you are interested in. Once you isolate this RNA, you then convert this RNA into cDNAs, and then these cDNAs are actually labeled with fluorescent dye. Now the condition A, you label the RNA with a fluorescent dye which fluoresces green, and the condition B, you label this RNA with a dye that fluoresces red. Then you mix these two RNAs together, and then you hybridize the microarray which we described in the previous slide. So this microarray actually contains, let us say for example this RNA is made from a mouse and you are hybridizing this RNA for a mouse microarray, and both these RNAs now hybridize. So let us now look at this microarray carefully. So you have all the genes which are actually present in the mouse genome, let us say about 30,000 genes, and each spot corresponds to one particular gene, and depending upon whether the gene is getting expressed in condition A, if this particular gene number 1 is expressed in condition A, then the green labeled RNA will hybridize to this, red will not. On the other hand, if gene number 2 is actually expressed in condition number B but not condition number A, then the red labeled RNA will hybridize to this. But if the gene number C is expressed in both conditions, then both RNAs will hybridize and you will ultimately get a yellow signal. So depending upon which gene is getting expressed, the corresponding RNAs, complementary RNAs will hybridize, and accordingly you get either a green signal or a red signal or a yellow signal, and that is what is shown here. Red actually indicates all those genes which are active in condition B. The green spots here indicate all those genes which are active in condition A, and the yellow spots here actually tell you genes which are active under both conditions A and condition B. Now sophisticated softwares are now available. You cannot do this analysis manually, so because they are all very highly microscopic regions. So you take this hybridization pattern and then put in using appropriate software, you can actually annotate which of these genes which are getting expressed in condition A or condition B or in both. For example, I have just given an example here. Say for example, I know exactly the region on the microarray here actually corresponds to a gene called QCR7, and this gene, if it is red, that means this QCR7 is actually expressed in condition B. Similarly, here is another gene, for example, you have YGP1 which is spotted, which is red here, which means that this gene actually expressed in condition B but not condition A, and so on and so forth. So using appropriate softwares and computers, you can actually identify which are the genes which are getting expressed in condition A, which are the genes which are getting expressed in condition B, or which are getting expressed in both conditions. So accordingly, you can actually also get the information on the fold induction, how many fold the RNA is more in one particular condition compared to the other, again using computational biological techniques. And once you have you can actually confirm that the microarray data you have got actually is valid. So you actually do an experiment. For example, if I want to really see whether the QCR7 is actually getting expressed only in condition B but not condition A, you again take the RNA from condition A and condition B and make a QCR7 probe and then do a northern blot analysis and actually show that yes, this data is correct. Similarly, I can do an experiment, actually take another probe. For example, if I am expressed here, I should validate again doing a particular northern blot, or you can also do what is called as a RT-PCR or real-time PCR, and so on and so forth. So this is basically how a microarray experiment is carried out. So you can see what this microarray actually given you the ability to analyze the expression of thousands of genes at any given time. So you can understand, or you can identify all the genes that are getting expressed in that particular cell at a given time, and you can actually compare two different conditions and make conclusions of what kind of expressed or what kind of genes may be responsible for that particular physiological response. If for example, when you add a hormone, if it is going to activate, what are all the genes which are activated by that particular hormone in that particular cell type. This can be found out by using these kinds of microarrays. So microarray became a very powerful tool to understand global changes in gene expression. I am just going to give a few examples just to tell you how actually a microarray data looks like. For example, I am interested in looking at the time course of serum stimulation of mouse fibroblasts. That means mouse fibroblasts which are normally quiescent, and as you know, if the mouse fibroblasts are to proliferate in cell culture, you require serum. Now as you know, serum contains a number of growth factors. So when you add serum to the fibroblasts, it stimulates cell proliferation. So a number of genes that are involved in cell proliferation should go up, and now I would like to know what kind of genes are activated first, what kind of gene expression genes are activated at the later time points. So I do a time course. So starting from minutes to hours, I now isolate RNA from different time points and do this microarray, and you can see clearly for very few genes are active when there is no serum, and once you add serum and then start doing time course, you can see a number of genes are getting activated, at the same time a number of genes are getting repressed. And I can now add to the microarray, and using those software data I can actually analyze and see what kind of genes are getting repressed when I add serum and what kind of genes are getting activated when I add serum. And then I can experimentally validate and then find out the physiological functions for these gene products. So I can now correlate, this is the mechanism by which addition of serum stimulates cell proliferation. These are the gene products involved and how these products actually interact with each other and then contribute to this cell proliferation phenotype. Similarly, here is another example where I am, let us say I am interested in gene expression profile of cells subject to oxidative stress at 10-second intervals. So I can give the oxidative stress and isolate RNA at 10-second intervals and then look at the gene expression changes that takes place because of this oxidative stress at different time points. And again you can see some genes are going up, some genes are going down, and one can look at these data and then make proper annotations and make certain conclusions. Here is another example. Suppose I made a mutation in one of the RNA polymerase II subunits, and I want to see how does this mutation of this RNA polymerase affect gene expression, because as you know RNA polymerase is very important for transcription. So again I take RNA from a wild type and I take an RNA from mutant and then compare these two different conditions and then see how this particular mutation of this particular subunit of RNA polymerase, what kind of genes are getting affected. And you can see these are the genes which are getting affected by this mutation in a RNA polymerase II subunit. So I can go on giving a huge number of examples, but the purpose is not to load you with information, but actually tell you what is the experimental approach that one follows if you want to understand global gene expression changes that are taking place. The microarray has become a wonderful tool to understand how gene expression changes can be measured at a global level, at the genome level. So this understanding gene expression changes is at the global level is actually known as expression profiling. Now what I told you is just one technique, that is how microarrays can be used for doing this expression profiling, but there are a number of other techniques also which are not as popular as microarrays, but they can actually complement certain defects of microarrays. These are called as SAGE, MPSS, TOGA and so on and so forth. But to lack of time I will not go into these other alternate techniques, but what I have done here is SAGE actually refers to what is called as serial analysis of gene expression. These are all the technologies which are also used to understand global changes in gene expression, and they are alternative to microarrays. So SAGE, massively parallel signature sequencing MPSS, total gene expression analysis, or TOGA. I have given the references. One who is interested can actually read these very nice articles and then try to order how these techniques also can complement the microarray technology for understanding global changes in gene expression. And these are all very powerful tools and gives you a huge volume of data and huge volume of information about disease phenotypes and so on and so forth. Okay. So what I have told you is to give a very comprehensive view about the importance of genomics, how genomics can be carried out by doing microarray experiments, and how the microarray experiments, I gave you a few examples of how information can be obtained on two different physiological situations, and how one can use the microarray data to understand global changes that take place in the area in gene expression. Now once the genomics, and once people started doing this kind of a genomics, and once people started mining what is called as data mining using these microarrays, people thought they can understand the biology of the organism. But it soon became clear that just by doing gene expression profiling we cannot understand everything, because ultimately we also need to understand how proteins function. As I said, once an mRNA gets translated to the protein can have pleiotropic functions depending upon its own state, depending upon its primary structure, secondary structure, tertiary structure, its interaction with other proteins, as well as post-translational modifications of proteins. All these things play ultimate role in ultimately giving a phenotype for a particular cell. Whereas gene expression profiling does not tell you what is happening to the protein. It can only tell you that these messenger RNAs are going up, these are going down, and you can then extrapolate and say these are the protein levels that are going up or going down. But what is the functional consequence of this? The protein levels may go up, but unless it is phosphorylated, the protein may not be active. So the phenotype of a phosphorylated protein may be different from a phenotype of a dephosphorylated protein. Similarly, glycosylation, or similarly unless the protein interacts with another protein. So it is not just enough if this protein goes up, another protein has to also is required for a particular phenotype, because unless these two proteins interact you may not get a particular phenotype. So it became very clear, just by doing microarray analysis or just by doing expression profiling you cannot explain all the things that are happening in the organism. So you need to also develop tools to understand global changes that are taking place at the level of proteins. And this analysis of global changes that are taking place at the level of proteins came to be known as proteomics. So genomics logically led to proteomics because it became clear it is not enough to just do expression profiling, you also have to look at the changes that are taking place at the protein level, and you need to have powerful tools. Just as the microarray, you also need to have powerful tools to understand changes that are taking place at the protein level. So let us now try to understand what is this proteomics and how proteomics has contributed to the understanding of various biological phenomena. So what is proteomics? Again, just like we defined genomics, proteomics is rapid identification of all the proteins synthesized in a cell or a tissue. So, just as genomics is rapid identification of all the RNA that is synthesized in a cell, here proteomics actually is the rapid identification of all the protein synthesized in a cell or tissue. So, proteomics is actually the study of a proteome, which is the protein complement of the genome, and proteomics is the study of protein expression, regulation, modification and function in living systems for understanding how living systems use proteins, because they are the ultimate workhorses. So, using a variety of techniques, proteomics can be used to study how proteins interact within a system or how proteins change under different conditions, including post-translation modifications and protein-protein interactions and so on and so forth. So, let us now discuss briefly what kind of techniques have been developed to do proteomics, how does one go about doing proteomics, and what are the applications of proteomics. The term proteomics was first coined in the year 1995 and it is actually defined as the large scale characterization of the entire protein complement of a cell line, tissue or an organism. And the goal of proteomics at that time is to obtain a more global or integrated view of biology by studying all the proteins of a cell rather than each one individually. This was the goal of proteomics. Why was proteomics necessary? As I just explained to you, I have now given a few more reasons. Having complete sequences of genome is not sufficient to elucidate biological function. So, it became clear just by sequencing genome after genome, just looking at the genome organization, you cannot really understand what is going on inside the cell, because ultimately you need to understand how these genes are getting transcribed and how the proteins are being made and how these proteins function inside the cell. Now, a cell is normally dependent upon several metabolic and regulatory pathways for its survival, and modifications of proteins can be determined only by these technologies. So, many a times how these metabolic or regulatory pathways are regulated ultimately depends on proteins, and in our previous classes we have clearly understood how various signal transduction pathways function and how protein-protein interactions and post-translation modifications of proteins play a very important role in various metabolic reactions as well as in various signal transduction pathways. So, it is necessary to determine the protein expression levels, post-translational modifications, protein localization as well as protein interactions, and only then we can get a comprehensive view of how exactly the genome is functioning and how the proteins which are transcribed from the genome actually are able to function and regulate their cellular processes. And remember, many a times it is ultimately the proteins which are the drug targets. So, unless you understand exactly how the protein is functioning, you cannot really develop novel drugs or they cannot be drug targets. So, understanding these proteins and how what kind of changes take place in the protein level became very important. So, what are the components of proteomics? So, if you want to look at very rapidly what kind of changes are taking place at the protein level, how do you go about? The key components involved in proteomics are: first, you need to have a very good protein separation technology, then you need to have what is called as mass spectrometry, and of course, you need to have a huge computing power to interpret the data, bioinformatics. So, proteomics combines protein separation techniques with mass spectrometry and bioinformatics, and together you can understand what kind of global changes are taking place at the proteome level. So, I will just briefly schematically describe here, although the whole proteomics is pretty complex, I try to simplify the whole thing, how exactly go about doing proteomics. This is the basic proteomic analysis scheme that I described here. So, suppose you have a protein mixture, let us say I have isolated proteins from a liver extract. I first have to separate these proteins. Now, normally we all do what is called as a sodium dodecyl sulfate polyacrylamide gel electrophoresis, but unfortunately the SDS page is primarily based on the separation of proteins based on their mass, because you neutralize the charge, all the proteins become negatively charged and they become reduced because you are adding SDS and beta mercaptoethanol. And many a times when you separate proteins on SDS page, you cannot separate proteins based on their charge. Suppose there is a protein which is differentially phosphorylated or differentially acetylated, you cannot separate these two proteins because their molecular weight is still more or less the same, and the fine difference between the molecular weight cannot be resolved on this one dimensional SDS page. So, if you want to do actually proteomics, you need to combine this SDS polyacrylamide gel electrophoresis with one another methodology that is actually based on separating proteins based on their charge. So, what you first do is, when you have a mixture of proteins, you first separate these proteins by what is called a two-dimensional gel electrophoresis, where the individual proteins are first separated by what is called as isoelectric focusing, where the separation is based on the charge, and once the protein got separated based on the charge, then you separate the proteins by their mass, or even what is called as a SDS page. So, by using isoelectric focusing and polyacrylate gel electrophoresis, you can resolve a number of proteins much better than either of them alone. So, this combination of these two techniques is actually called as two-dimensional gel electrophoresis or 2DGE. So, combination of isoelectric focusing combined with polyacrylate gel electrophoresis. So, once you have resolved the individual proteins based on the charge as well as mass, you can now take individual spots, individual proteins, and break them into peptides using enzymes like trypsin, and then this peptides is fed into an instrument called mass spectrophotometer. We will not go into the functioning of the mass spectrophotometer because that is a totally different thing, but basically in the mass spectrophotometer the proteins get ionized and the mass of each one of these peptides is analyzed. And so, what you ultimately have, the data that comes out of a mass spectrometer is the mass of various peptides that have been generated for a particular protein by the trypsin digestion. So, you have a series of mass of peptides depending upon the number of trypsin cleavage site in that particular protein, you get a whole mixture of peptides with different masses. Now, there are actually databases, what is a very popular database called as mascot, where the predicted or theoretical peptide masses of all known proteins are available. So, you take the protein peptide masses that you have got from your experiment, and then if you now feed into the database, it will now compare the data generated by your experiment with the predicted peptide masses of all the known proteins, and then match and then tell you this is the likely protein that is present in your sample. So, it will identify a protein based on this kind of a comparison. So, this is basically is the basic proteomic analysis that one carries out. So, let us just spend a couple of minutes to understand how exactly you do a two-dimensional gel electrophoresis, because that is the key technique. So, as I told you, the two-dimensional gel electrophoresis basically is done first by isoelectric focusing followed by SDS page. Now, in the isoelectric focusing, basically you have a gradient from various pH, it can be from pH 3 to pH 10, or if you want to separate proteins at narrow range, you have pH strips where you can start from pH 7 to pH 10 and so on and so forth. So, depending upon the pI that you are interested in, you can use different kinds of pH strips. Now, then you apply your protein sample and pass current from negative to positive charge. Now, at time 0, before you apply the current, the protein will be all over because they are under various pI. Now, once the current is applied, the proteins start migrating under the electric field, and they will stop migrating when they reach their isoelectric point. So, here the proteins get selected in the isoelectric focusing depending upon their pI, and you get various proteins that get separated based on their pI. So, this actually is called as a first dimensional electrophoresis, where proteins have been separated based on their pI. Now, once you have done that, you now take this strip and then now subject it to SDS polyacrylate gel electrophoresis, where now the proteins which have been resolved based on their pI, now they move in the electric field and now they get separated based on their mass. High molecular weight proteins move less, migrate less, and low molecular weight proteins move further, and so ultimately what you end up at the end of this two-dimensional gel electrophoresis is proteins which have been resolved based on their charge as well as the mass. So, this is the schematic of how exactly you do the 2D you get. You have mixture of proteins which have now been first separated on the IEF based on their charge, and now when you subject it to the second dimension SDS page electrophoresis, they get resolved based on their mass, with high molecular weight proteins at the top and low molecular weight proteins moving the farthest. This is how basically a 2D gel looks like. You can either stain this gel by Kumasi blue, or you can do silver staining, or if you have used the radiolabeled proteins, you can do autoradiograph proteins and then pick up the radioactive spots. So, you can see each dot here represents a particular protein. Okay. And so, once you have separated the proteins, now how do you identify these proteins? These proteins are actually identified. Here is, for example, I have given you one example of what you do by two-dimensional gel electrophoresis. Let us say, for example, this is the protein mixture from a control sample, and this is a protein mixture from a experimental sample. And as I mentioned in the case of microarray, it can be any two different physiological situations. Control means unstimulated or stimulated. It can be quiescent cell versus proliferating cell, without ligand, with ligand, any two different physiological conditions. And you can see here all the proteins are more or less same, but you can see there is one particular protein which is now present in the experimental sample, but is absent in the control sample. So I now make a conclusion that here is a particular protein which is getting induced, which is present only in these experimental conditions, but not in the control conditions. Now we want to identify what this protein is, and you can see the power of this technique. You have not purified any protein, you have not separated any proteins, you have simply taken a whole extract or the entire protein complement of a cell or a tissue, and under two different conditions you separated all these proteins, and you can already see there are differences between the two. There are protein profile which is different. Now let us see how you proceed further. So once the proteins are separated by two-dimensional electrophoresis, now how do you identify the protein? So for example, in this case, now I want to now see what is this protein that is actually appearing only in this particular condition, but is absent under this condition. What do? I now excise this gel piece which contains this particular protein sample, and then subject to what is called as a trypsin digestion followed by mass spectrometry. So as you know, trypsin always cuts after a basic amino acid, either lysine or arginine. So you take this gel piece containing this trypsin and subject to what is called as a in gel or in situ trypsin digestion, and then you take these peptides and then put it inside a mass spectrometer. So these peptides, when they are introduced to mass spectrophotometer, as I said, I explained earlier, these peptides will be ionized, and the mass of each one of these peptides is determined by the machine, and the values that are coming out of this machine can now be fed into a database, and this is what is called as a peptide fingerprinting. Now, so depending upon the mass of the peptide that are generated and comparing the mass of these peptides with known databases, you can actually identify what this protein is. Now there is one more advanced technique, which is what is called as a tandem mass spectrophotometry or MSMS, wherein once the peptides have been generated and once the peptides have been characterized, and you can now ask the machine again to take any particular peptide that you are interested in, and this peptide is again subjected to one more mass spectrometry, wherein these amino acids are cleaved from these peptides, and these amino acids are again ionized, and again based on the mass one can now identify exactly what is the amino acid sequence of that particular peptide. So you are doing two mass spectrophotometry twice, therefore it is called MSMS or tandem mass spectrophotometry. So first you are looking at the mass of the peptides, then you can pick up peptide of your interest, again subject it to the mass spectrophotometry, and you can identify what is the actual amino acid sequence of this particular peptide. So it is a very powerful tool. Mass spectrophotometry became a very powerful tool, wherein you can actually identify a particular protein from a crude sample, from a tissue or a cell extract without any purification. So this approach is actually called as a bottoms approach proteomics, wherein proteins are identified at the peptide level. So peptide mass fingerprinting actually uses the masses of the proteolytic peptides as input to such a database of predicted masses that would arise from the digestion of a list of known proteins. This is what I explained just now. So if a protein sequence in the reference list gives rise to a significant number of predicted masses that match the experimental values, there is some evidence that this protein was actually present in the original sample. So the peptides which are generated, the masses of the various peptides that you have generated experimentally, you now compare against the predicted triptych digestion pattern of all known peptides, and if this peptide fragmentation matches, then you can reasonably confident that your protein is likely to be what is depicted from the database. So in the tandem mass spectrometer or MSMS, the sample proteins are first broken to short peptides using trypsin, separated using chromatography, and then they are sent through one mass spectrometer to separate them by mass, and then peptides, once they are separated by mass, the peptides having a specific mass are then again typically fragmented using collision induced dissociation and sent through a second mass spectrophotometer or mass spectrometer, which will generate a set of fragment peaks from which amino acid sequence of a peptide may be inferred. So we are doing mass spectrometry twice, one first to generate the peptide mass and then to actually generate the amino acid mass, and by comparing these things you can actually identify using appropriate software, you can actually identify the sequence of the peptides and then infer what the protein is. So instead of doing this, is a mass spectrometry became a very powerful tool to identify proteins from crude samples and became an very good alternative for the classical amino acid sequencing or protein sequencing. using Edman degradation and so on and so forth. So, this is a schematic I have shown here, again I will not repeat. Basically take a protein subject to trypsin digestion, take the tryptic peptides, put in a mass spectrometer, you get various peptides, and then you take these peptides and you have databases of predicted protein sequence which are where the predicted tryptic peptide mass are given. By comparing with the theoretical probable, you will identify what is their protein likely to be. So, mass spectrometry became such a powerful tool that the people who actually developed these techniques, John Fenn and Koichi Tanaka, were actually awarded Nobel Prize in the year 2002. The citation actually states that Nobel Prize in Chemistry 2002 was awarded for the development of methods for identification and structural analysis of biological macromolecules, one half jointly given to John Fenn and Koichi Tanaka for their development of soft desorption ionization methods for mass spectrophotometric analysis of biological macromolecules. So, the mass spectrometry became a very powerful tool for identification and characterization of protein molecules, and it became a fantastic tool to understand how proteins vary from one particular situation to another situation, became excellent to a normal versus phenotype or two different experimental conditions to identify proteins by using this kind of a mass spectrometry techniques. So, here is for I have shown a couple of examples. You can actually compare the proteome of undifferentiated cell versus a differentiated cell, or you can compare the two proteomes of uninduced versus induced. It can be a hormone, it can be a growth factor, and so on and so forth, or it can be normal versus mutant, where a transcription factor or a signal specific component of signal transduction pathway has been mutated. Now, want to find out what kind of proteins are getting affected by these mutations, or it can be minus hormone or plus hormone. You can see what kind of proteins are getting synthesized when I add a particular hormone. Sky is the limit. You can compare any two different physiological situations and get the variations in the proteome. And so far I have talked about analyzing in one gel only one particular condition. So, if you want to compare two different conditions, you have to run two gels. Now you have what is called as a two-dimensional differential gel electrophoresis or 2D DIGE, wherein here you do not have to run two gels. You can mix both these. You can take the proteins from two different conditions, mix them together, and you can get, by running a single gel, you can infer the data. This is now similar to what we did in the case of microarray, where you label the RNA from one condition with a green dye, another condition from a red dye, mix them together, hybridize to a microarray. The same way here, you can now take the proteins and label them with the dye 1, which fluoresces differently, and take the other condition 2 and label those proteins with another dye which fluoresces differently, mix them together, run the gel, and then you can see identify these differentially fluorescing proteins. So, the two-dimensional gel electrophoresis or 2D DGE, you do not have to run two different gels. You can run it by running a single gel. You can find out the differences in the proteomic pattern. And many a times now you can also induce a standard which should not change. It basically tells you that your experiment is all right. So, you have actually red label proteins, blue label proteins, and when you mix them together you get a combination. This is how a typical gel looks like. You have raw blue dots, red dots, and yellow dots, so on and so forth. So, by running a single gel you can get information about differential expression of the proteins. Now, there are a number of variations of these proteomic techniques that are now available. So, what I told you is only about MALDI or mass spectrophotometry based on MALDI or MSMS. You have what is called as a liquid chromatography mass spectrophotometry or LCMS, where instead of separating the protein on a two-dimensional gel by IEF and SDS page, you can separate the proteins based on a HPLC by liquid chromatography system, and then these proteins are fed into mass spectrophotometry. So, combining the mass spectrophotometry with the liquid chromatography technique is known as LCM. So, you can either separate proteins based on SDS page, or you can separate proteins based on liquid chromatography systems, and ultimately you can combine them with mass spectrophotometry and get the data you want. This is called as a electrospray ionization mass spectrophotometry or ESIMS, again where intact proteins are ionized and then introduced into a mass analyzer, and this approach is actually referred as a top down approach for the strategy of protein analysis. There are a number of databases now available. The most widely used database for that is, once you generate the peptide fingerprint, that means once you got the masses of the various peptides, you can take this and then go to these databases and feed these numbers into those databases, and the software will now compare your mass values with the predicted triptych patterns of all known proteins and give you the identity of your protein. So, a number of such databases are now available. So, what are the applications of proteomics? I am just going to give you a few examples. Just I mentioned in the case of genomics, you can do proteomics for protein mining. That is, you can catalog all the proteins which are present in the tissue, cell or organelle. So, just as you have a gene expression signature using DNA microarray, you can get the protein signature, what all the proteins which are present in a given situation in a given cell or a given tissue by doing proteomics. And as I mentioned, you can do differential expression profiling, identification of proteins in a sample as a function of a particular state. It can be differentiation, different stages of development can be compared, disease state, normal state versus disease state, response to a drug or stimulus, you name an example, can be done by doing proteomics. You can do network mapping, identification of proteins in functional networks, biosynthetic pathways, signal transduction pathways and multi-protein complex. I will just give one example as we go along. Mapping protein modifications, again when you run a 2-dimensional electrophoresis, you can actually separate the proteins based on their post-translation modifications because the charge will vary. So, you can characterize the post-translation modifications such as phosphorylation, glycosylation, acylation, etc. So, I just give an example of a differential protein expression. For example, by identifying the proteins present in a undifferentiated versus differentiated, you can actually put them together and find out which are the proteins which are unique to differentiated cells, which are the proteins which are unique to undifferentiated cells, and what are the common proteins which are present in both differentiated versus undifferentiated states. So, lot of data can be obtained. Clinical proteomics. No, serum is the most important source for or most easy way of identifying, very versatile method of identifying a disease phenotype. And suppose there are certain serum proteins which can serve as biomarkers. So, you simply collect the serum and you can feed these entire serum proteins by to separate them by 2-dimensional electrophoresis or LCMS, subject them to the mass spectrophotometry and then see whether any protein is missing or whether a new protein is appearing, and based on that, by identifying the protein you can correlate whether it can lead to any particular disease phenotype. So, clinical proteomics is another very major area that came out of the proteomics, understanding the serum protein pattern, identifying protein biomarkers in various serum. Study of protein-protein interactions. As I said, ultimately either it be a signal transduction pathways or metabolic networks, it is the protein-protein interactions ultimately are responsible. So, suppose I want to know, let us say for example a protein called S14, what are the various other proteins which are interacting with the S14. So, I can take this S14, express it as a GST tagged protein, couple it to the affinity column and then pass a cell extract through this column, and then by using different stringency I can see what kind of proteins are interacting with the S14, and then I can simply take this again do mass spectrophotometry and identify what are these various proteins which are interacting with S14, and based on this I can develop what is called an interactome map. Interacting network, what is called a protein interaction networks I can develop, and then make some conclusions on how S14 interaction with all these proteins can contribute a particular pathway or a particular phenotype. I just give an example here how for example a protein called as DISC1, by doing this kinds of proteomes, what kind of proteins are actually interacting with this DISC1 and how this DISC1 can this protein plays a very important role in schizophrenia. So, by understanding what kind of proteins are actually interacting this DISC1 protein, one can infer on what is the basis for the schizophrenia and how this protein may be actually involved in schizophrenia and so on and so forth. So, this is just a cartoon that actually tells you various applications of proteomics. Expression profiling can identify post-translational modifications, protein-protein interactions, structural proteomics, functional proteomics, proteome mining and so on and so forth. So, following genomics, proteomics became a very versatile tool for understanding how proteins function inside the cells and how protein-protein interactions can ultimately contribute to metabolic networks and signal transduction pathways. Now, so far we talked about DNA microarrays. Now, there are coming up what are called protein microarrays. So, proteins are actually being spotted on these glass slides, and using this kind of a protein microarrays, one can study antigen-antibody interactions, one can study protein-protein interactions, you name it, small molecule protein interactions and so on and so forth. So, suppose you identified a protein as a drug target, you can simply, and suppose you have a chemical library of various compounds, you want to see which kind of a drug molecule actually binds to this protein, you can simply spot this protein and pass various drug or candidate drug molecules and then see which drug molecules actually binds to this protein with highest affinity. So, protein microarrays, or we can study antigen-antibody interactions, ligand protein interactions and so on and so forth. There is an excellent review article to understand protein microarrays and their comparison to DNA microarrays in current opinion chemical biology and methods in molecular biology. I suggest one can, by reading this, you can understand more about the potential of protein microarrays. There are newer techniques coming up. There is a very sophisticated technique called as surface enhanced laser desorption ionization mass spectrometry or CELD-MAS, wherein for example you can take the protein array and for example I want to identify serum proteins which are interacting with the particular protein. So I can now apply a serum sample to this protein chip, and then by using different wash buffers of different stringency I can wash off. So by increasing stringency, as you can see, the number of protein molecules interacting with this protein chip can be minimized, and you can see what are the molecules who are interacting at the highest stringency versus what are the molecules interacting at the lowest stringency. And again I can now take off this by actually energy absorbing molecules are added to the retained proteins, and following laser desorption and ionization of proteins and time of flight mass spectrometry you can actually remove these proteins and then generate various peptides of different mass, and you can actually identify these proteins that are interacting with the interaction. You can identify what kind of serum proteins are interacting with your protein of your interest. So I think what I basically told you is about the potential of DNA microarray and potential of protein arrays, how you can do genomics, how you can do proteomics, and what is the potential of genome analysis and proteome analysis, how it has contributed to the understanding of various biological phenomena, and has really revolutionized the way biology and medicine research is carrying out in biology and medicine. I have given a few references here. One can actually go to some of these websites and gain more information about how DNA microarrays are being made and how DNA microarrays are being used. Affymetrix, Stanford University, and a number of other websites one can visit. There is actually now a separate organization called Human Genome Organization, HUGO. One can go to the website and you can get fantastic amount of information of how human genome analysis, sequencing and genome analysis has contributed to understanding of biology and medicine. There are very nice review articles in Scientific American on proteomics. One can actually download this as a PDF article by going to this particular website. It gives you how proteomics is expected to change the way biology and medicine research is being done in these areas. There are some very nice review articles I have listed here. Just as we have a human genome organization, now we have human proteome organization, and again you can go to the website and read some of these articles. It gives excellent information, and ultimately one must read these two Nobel lectures by Fenn and Tanaka, who explain how the development of the mass spectrometry has revolutionized biology and medicine. I think I will stop here. I think this is the last slide where I actually, this is a database called as Kyoto Encyclopedia Genes and Genomes, which actually talks about various metabolic pathways. All about 240 organisms, 20,000 organic specific pathways containing about more than 700,000 genes have been compiled together, and networks and pathways have been built, and very useful to go and then look at some of these pathways, compare the various pathways across organisms and so on and so forth. I think I will stop here.